To get started, welcome to the Ohio State University Soil Health Webinar Series for 2026. Today, we are focusing on soil biology, the living engine of soil health. So some housekeeping. Today, you will earn, if needed, one CEU in soil and water conservation. It's offered today. There's certification information will be at the end of the program. I believe we have a QR code that you can scan at the very end of the program. We'll make sure we keep that up on the screen near the end of the program long enough so all of you can take your phones out and scan that. The session will also be recorded. It's available at agcrops.osu.edu. And throughout the program today, feel free to submit your questions at any time using the Q&A chat feature at the bottom of your screen. You can also email questions to Asmita, and here's her email address in front of you. And what we will do is we will use the chat feature and poll questions to get feedback from you likely afterwards. And so with that, I'll move forward. And what we would like you all to do, all the participants online, is to share with you or share with us where you are from. And if you can type your response in the chat to all panelists and attendees, that would be fantastic. This way we have an idea if you're from Ohio or from Florida or from Brazil or wherever you may be from. So I'll give you a minute or so to do that. And we appreciate you entering that information. Okay. While you're entering that, I'd like to introduce today's panelists. With us today are Dr. James White and Dr. Carlos Pires. And Dr. James White is professor of plant pathology at Rutgers University. Dr. White obtained his MS in mycology and plant pathology from Auburn University in Alabama and his PhD in mycology from the University of Texas, Austin. Dr. White specializes in symbiosis research, particularly endophytic microbes. Recently, Dr. White and collaborators have been examining the role of intracellular bacteria in modulation of plant development and nutrient acquisition. And our second panelist is Dr. Carlos Pires. Carlos is an assistant professor of soil health at North Dakota State University. He obtained his MS in soil science from Federal University of Santa Maria, Brazil, and he earned his PhD in agronomy from Kansas State University. Dr. Pires' research expertise includes regenerative agriculture, soil health, nutrient management, and applied soil biology. Much of his research is based on-farm research, providing data-driven recommendations to help farmers enhance the profitability and sustainability of their operations. And so with that, what I'd like to do is turn it over to Dr. White for his presentation. And I'll stop sharing and James, it's all yours.
Thank you, Jim. I appreciate that. First, I want to say thank you for inviting me to participate in this webinar with all the speakers. I mean, with Carlos, Dr. Pires, and all of the attendees. So I really appreciate being here, being able to talk about the work that we do. What I'm going to talk about predominantly are how plants use endophytic microbes these are microbes that go into their tissues, but also how they use soil microbes in a process that's come to be known as rhizophagy, or rhizophagy, depending on where you're at and how you pronounce it. But so I'm going to share slides now, hopefully. And let me do that. Yeah, hold on. Okay, is this working?
Yes, it's not in the presentation mode.
See it? Hold
Yeah, if
on.
you want to turn it to the present. Yep, there you go. Thank you.
We got it. Okay, good.
Yep.
Good. So basically, this is how plants use soil microbiology. And you've likely heard about endophytes, and these are just any microbes that go into plant tissues. And this is actually an image that shows a fungal endophyte in a grass plant. And the blue line is the fungus in there. But endophytes, internal phyte plant. Any microbe that goes inside a plant that doesn't cause disease. So it's non-pathogen. They could be bacteria. Like this is a bacterium. You can see this is actually in a little weed. It's in a weed seedling. These are the cells here. You can see I'm kind of circling the cells. This is at a root tip. And these little blue dots in there, those are the bacteria, bacterial endophytes. So they endophytes can be bacteria. They can also be algae in some cases. Okay, basically what plants are doing is they're cultivating microbes in association with their meristems. And this is where they give plants, plants will secrete out exudates and attract microbes and then cultivate them and actually take them into the cells at those meristem areas. And the meristems are dividing areas. So the root tips and the shoot tips, they have meristems. Okay, this shows a root tip of a grass. See to the left, this is a root tip with actually bacteria, and you can see the brown color there. That's where the bacteria are. The plant is cultivating those bacteria right there at that root tip. You also notice there's hairs there. The bacteria cause development in the plants. And so without any bacteria there, you have no root hair development. The bacteria are necessary to have proper development in plants. Look to the right. This is where a root tip of a grass, where we remove the microbes from the seed so that there's very, very few microbes associated with the seed. Then we germinate them, and you see there's no hairs. You see also no bacteria cultivated there, being cultivated on the tip. So plants are cultivating these microbes, and they're taking them in, and they're using them for development and other things. Okay, what are these microbes? Okay, they're soil microbes. Basically, all of them are soil microbes that the plant will get from the soils, and then the best ones they will put actually on the seeds when the plant produces seeds, and so they'll go on and in the seeds, and so they'll be seed vectored. And this is a grass, germinating grass. You can see the root coming, grass seeds, you can see the root coming out. Well, there's microbes actually around that root. There's also microbes, you can kind of see a yellow coloration there. Several microbes, there's a little community of bacteria and fungi that are vectored on those seeds. So you actually want to be very careful in terms of application, in terms of sterilizing seeds. You don't want to do things to seeds that will affect the microbiology on those seeds. Okay, so you have to be careful with doing things like removing husks from seeds because the microbes are carried on husks. Any kind of sterilization that you do of seeds, you want to be careful about that you don't damage the community of microbes there. Now they can get them back, no doubt about it. If you have a good, healthy soil, they can get those seeds back. But they're certainly early in development. Over the past 15 years or so, we've understood that there's something going on where plants are actually absorbing microbes into their cells. And that's come to be called rhizophagy. Rhizo for root, phagy for eating. Okay, in which plants will actually absorb microbes into their cells. And that was done. This was actually some work done out of Australia, and these were two of the investigators on that study. But this is from their paper. And what they did is they labeled the yeast and bacteria with green fluorescent protein. They gave it to plants, and then they could track it using fluorescent microscopy. And so they could see it inside the plant. So they knew that the microbe they put on is the microbe that got absorbed and went into the plant. And then it vanished. The microbes vanished after a few days. And so this originates the idea that plants are actually getting nutrients out of these microbes. They published that in a research article in a journal called PLOS One. And you can see title, Turning the Table, Plants Consume Microbes as a Source of Nutrients, and that was in 2010. It's open access. You can go and see that article anytime you want, free. There's no paywall on it. Well, the bacteria, plants absorb bacteria, and it shows here to the left, you can see bacteria. They take them into their tissues, and you can see them here in actually root hairs. These little brown dots, those are the bacteria that the plant took in, that plant took in. They can take yeast in. This is yeast cells here, little fungus, right? And it actually is taken in. This is in a clover. You can see the root hair here. You see these little bitty dots in there. Those are bacteria inside the plant, and you can see them coming out of those root hairs right there. You can see small ones when it first come out, and you can see big ones later. So they come right out. So it's kind of a cycle, actually. The algae can go in. This just shows an alga in a root hair there. And oftentimes the algae will bring in bacteria when they get absorbed. So there's a whole, if a plant takes in algae, it also takes in some bacteria. We found it's a cycle, and what happens is the plants at the root tips secrete exudates, and these are sugars and organic acids and compounds that act as attractants to soil microbes. Soil microbes of different types respond to that, and they're cultivated at the tip, and then the plant will take them into the cells. Okay, at that, and exactly how that happens, we don't know. There are some hypotheses there, but we don't have to dwell on that because there's uncertainty there exactly how. We just know that they go in because we can see them going in, and we can see them after they're taken in. Okay, then once they're in, the plant will use reactive oxygen, take oxygen, a very potent form of oxygen, superoxide, strip off the cell walls of the bacteria, oxidize off the cell walls, and we think take nutrients in the process of oxidizing cell walls. And some of the bacteria break down themselves too, but the cell walls are taken off, they become protoplasts, and then the plant replicates them inside the plant cells, particularly inside the root hairs. Okay, and so if you look over here, you can see these are the root hairs. And if you look at the tips of the hairs, you see little balls. Those little balls are actually bacteria that are coming out. So the bacteria go in at the tip or just behind the tip, just behind the meristematic area, and they end up going into the hairs, and then they come out of the hairs. And then once they come out, they'll reform their cell walls. Okay, and so the plant actually will give them a little bit of exudates out there so they can make new walls. Then they can go back to the soil and acquire new nutrients. And so you see this cycle. So we called it a rhizophagy cycle, microbes that go around. And this is a symbiosis, no doubt about it. It's a symbiosis. It's a kind of a non-specific symbiosis. I would call it a process that plants engage in order to get microbes that it can utilize, taken into the cells, and then process them, use them for development, use them for nutrients, use them for causing plants to upregulate other kind of like oxidative stress genes and so forth. Okay, all of this rhizophagy cycle happens at root tips. Root tips. So all of these is a corn root. You see those little branches there? Those are all the branch roots. And so rhizophagy is happening at every one of those little root tips. So if you imagine a corn plant with many, many branch roots, many, many branches coming out, there's a lot of rhizophagy happening all over that. Okay, this shows one. You can see the root tip, branch root, and you can see all the bacteria cultivated right there, right behind that meristem, right in that zone. And that's also where they take those bacteria into the cells, right in that zone. Okay, and this actually shows this is stained for bacterial walls using, this is another grass stained using crystal violet, which stains bacterial cell walls, the protein in cell walls. And they come in at the tip right over here. And behind the tip is where they are hit with reactive oxygen superoxide to remove the cell walls. And so the cell walls are coming off here. And so if you look down here, this is kind of an insert. It's irregular because that's where the cell walls are coming off. And we think the nutrients, many of the nutrients are carried on those cell walls. So metals and things like that, micronutrients and different compounds are actually attached to the cell walls, and cell walls have protein in them as well. So that can all be oxidized, and that becomes nutrients for the plant. At least that's what we think. Here you can see the image. This is a close-up of it. You can see the brown coming off. I mean, the dark purple. Those are the cell walls that come up inside the plant cells at that point. What happens when the plant takes the cell walls off? Here it looks this is a bacterium. You see the rods with cell walls. When the cell walls are removed with superoxide, then they just become these little spherical structures inside plant cells. And they look like organelles, but they're actually what are called protoplasts, or L forms is a fancy name for it, but it's just protoplasts. They're wall-less bacteria. Well, these are important for development. I mean, for example, the microbes trigger root hair elongation. This is a grass root. A grass root. This is really a Bermuda grass root where we removed all the microbes. And you can see there's no hairs there. There's no hairs at all. Without microbes, no hair happens. Here's when we added the microbes back in. This is the same plant. Okay, same seed, same Bermuda grass. Different individual Bermuda grass, different seed, but we put bacteria, we removed it, then we put it back on, and here you can see the hairs form immediately. It makes hairs. And if you look at the hairs that are forming on those, you can see those little brown dots. Those are all the bacteria. Those are all the bacterial protoplasts inside those. They get full. And what we think is that they're producing hormones, and those hormones that are producing things like ethylene and nitric oxide are what's responsible for the growth of those root hairs in plant tissue. Okay, and this just shows that the microbes will accumulate into tips of those hairs, and in a mass they'll produce hormones, and like ethylene, and we can visualize ethylene in there, and we think we can visualize nitric oxide as well in there. And both of those act as elongation factors for root hair development, but also other parts, other cells will develop also. Well, the root hairs actually are processing structures for soil microbes. And here's an example of a plant that grows mostly without soil. It's a little sedge, and it's growing on these limestone coral formations in one of the Caribbean islands, Bonaire, right, in the Dutch Antilles. But if you look at we stain it for reactive oxygen, and you see this is a root hair below of that plant. You see these little brown areas on the edges where the arrows are indicating. That's where the bacteria are. Okay, but that's kind of doesn't tell you exactly what's happening. If you look at the one above, this is a living cell. This is the movie looking at a cell and what's happening in that cell. And you can see now the little shadows going around. Those little shadows are the bacteria that are being processed, cycled around and around and around and around and around. The plant is actually breaking up these microbes into little bits, to little individual colonies and individual cells. Here, this is a still of that, and you can see a cluster. You see some are big and some are little. These came from a single cell, a single bacterial cell inside the root. So the plant, in moving it, the plant is breaking them up. The plant is making more of the microbes that it takes in. It's replicating those microbes, and then it's going to put it out of the root hairs. When it forms those root hairs, you can see they're accumulating. See the little white things here? Those are the bacteria in the tip. They accumulate in the tip. And then at some point, they trigger growth. And when that root hair grows, inside that, the root hair cell grows, and it pushes those bacteria out into the soil, back into the soil. And so that's what we were seeing in the previous slide. But here you can see these hairs. Some of these hairs get fat, full of microbes. And look at the microbes when they came out. Okay, so they come out, and then the plant will put some exudate out there to feed them, to actually give them some nutrients so they can reform their cell walls, and any flagella essentially become capable to live in the soil again. So it's really the plants are processing these microbes. They're farming or ranching these microbes. So plants, like we farm plants, plants farm microbes. Okay, and here you can see actually bacteria coming out of the tip. There you can see them in the tip. They were ejected there. You can see them there. Okay, and basically the process is in the process of rhizophagy cycle, plants will get nutrients, okay, from those cell walls or from breaking down some of the microbes. They also get nutrients from soil activities of microbes, right? And they can absorb whatever the microbes are doing in the soil, phosphorus and so forth. They can get plants become stress tolerant because they're oxidatively interacting with microbes in there. So they get that stress tolerance. They upregulate their own stress tolerance genes, their own antioxidants and so forth, and phenolics and antioxidant chemicals. So they become more resistant to stress, any kind of stress, when they have microbes. They also, these microbes go into the soil and they'll affect fungi, fungal pathogens like fusarium, and they'll reduce their virulence. They'll colonize them, cause them to be less virulent or non-virulent. And sometimes the pathogen will become an endophyte or a symbiont of the plant. And the other thing that they'll do is they help plants to adapt to the environment, to the particulars of their environment. So plants will adapt, they'll absorb, and they'll have symbiosis with microbes that help them survive in whatever environment they're in. Say it's a high salt environment. Well, they'll get microbes that help them with that salt tolerance and so forth. So that is, these microbes are actually doing a lot for plants. And I think that's all that I have to talk about. This is in a book, okay, Teeming with Bacteria, okay, by Jeff Lowenfels. Okay, and you can get that. You can get that if you need that. If you want a book that says a little bit more about that. And I think that's all. Let me stop the share. Stop the share. I hope I didn't go over. I'm sorry if I did.
No, you didn't. Wow. I'll speak on behalf of everybody online. I think you probably could have went a few hours and it would have been fascinating. It was fascinating for sure. I need to have you come talk to my class next fall. I'm not kidding. Um, Thank you, Dr. White. Excellent presentation.
You're welcome.
Let's move over. Dr. Pires, the floor is yours.
All right, let's go through the screen sharing thing. Okay, I think you can see it. Can you?
Yes.
Yes, we can.
Okay, awesome. Now I'm using multiple screens here. Now I'm trying to find my screen. Just 1 second here real quick. Well, I may have to stop sharing and share again. Just 1 second. Okay, let's see. Okay, I think it's working now. Is it?
Yes.
Awesome. Okay. Well, just want to say thank you for the invitation to speak this morning. It's really good to, I'm in Central Time, so it's right now it's 7:23, and it's really good to just wake up in the morning, get a cup of coffee, and talk about soil health. So appreciate the opportunity to do that. And so what we're going to do today is going to be a little bit different, and it's really hard to follow Dr. Wright. But we're going to talk a little bit about the applied biology. So what happens to biology when our farmers implement the soil health principles and soil health practices? And I'm gonna have to skip a few of these slides for the sake of time. But we usually say that soil health is about the physical, biological, and chemical component of the soil, right? And sometimes it's really hard to put names to this physical, chemical, and biological. So one of the things that we have done in the past was to create what we call the soil health holy trinity. And then when you put the name to these properties, it makes a little bit more sense. And you can also see that biology actually affects or has a saying in most of the other properties of the soil. So here's just a quick example of this biological component, like microbial activity, the physical component, soil structure, and the chemical component, organic carbon. So looking at this way, you can clearly see the relationship between these three components of soil health. Now, a good way of looking into the biological component, and I just wanted to bring the soil food web to today's presentation because we often forget about some of our biology. So we talk a lot about soil microbes and the microbiome and genome sequencing and all that, but we usually forget about our macrofauna, right? So we have microorganisms, they do a lot for soil health as well, and that's the reason I wanted to bring this to today's presentation. So to remember about the earthworms, beetles, and so on. Now, I like this figure because it puts the size of these organisms in perspective, the soil food web or the biology of the soils. And they all have different functions. And if you look into let me get the laser pointer here, but if you look into the sizes here, so earthworms will measure anywhere from 2 to 8 millimeter, while bacteria will be less or 1 or 2 micrometers. So there is a big difference in terms of size. And because of that, they have different functions in the soil. And we still have fungi, nematodes, protozoa, mites, and so on. So all different sizes and all with different functions in the soil. And of course, this is oversimplified, but these are some of the roles of soil microbes and soils. And before getting into some of these roles, so we know about 100,000 species of soil fungi, of an estimated 5 million species, and about 5,000 identified species of bacteria compared to an estimated 1 billion species of bacteria that we have in the soil. And it's one of the reasons why we do a lot of research with microbiome and the role of these microbes, because there's still a lot to be figured out. But just simplifying a little bit of the role of bacteria, so they decompose nitrogen-rich material, so which means that type of residue with a low CN ratio, so they love to decompose organic matter as well, and they help to bind soil particles into aggregates. If you look into fungi, and they are well known for improving soil aggregation because of their hyphae. They decompose high CN ratio material, so which means material with more lignin. So if you have to put in perspective, so fungi would decompose grasses, for example, where bacteria would have a harder time doing that. Arbuscular mycorrhizal fungi, so AMF, really important for plant and to enhance plant nutrient uptake. Also really important for aggregation and protozoa, so they are important decomposers and they regulate microbial population as they consume bacteria and release nutrients into our soil system. So another really important aspect of this biology, and it's becoming more and more common in soil health assessments, are soil enzymes. And I just wanted to bring a few examples here of the ones that are mostly measured by commercial labs and also in research. And so we've done a meta-analysis in the Northern Great Plains, and we found, of course, beta-glucosidase is the most measured enzyme in research. But they have different functions in soils. So beta-glucosidase will help in the carbon cycling process. Acid phosphatase or alkaline phosphatase will be linked to the phosphorus cycle, aerosulfatase to sulfur, and then NAG, which I just don't have the courage to pronounce that, so we'll go with NAG in that case. But they all have different roles in soils and are part of our biology. But as I said before, so mesofauna and macrofauna, they are also important. We often forget about that in academia. There's very few research, and there are very few speakers actually bringing attention to the mesofauna and macrofauna when it comes to soil health across the country, mainly soil health and extension soil health, I should say. And honestly, farmers are always talking about earthworms, right? They're always talking about how many earthworms they can find in their soil, and because of that, their soil is healthy. But I think it's a little bit of forgotten by the research community. But the mesofauna are really important for microbial regulation because they graze fungi and bacteria, so they break down residue, really important for nutrient cycling too. Macrofauna like earthworms or termites and ants and beetles, They are soil engineers. They are important for bioturbation and soil mixing, and of course, pore formation, which is one of the most important things when it comes to soil health management. When we are transitioning from a conventional system to a no-till system or reduced tillage system, so one of the things that we really need is that formation of biopores. And then I just have an animation here so you can see. So these are soil cores from Kansas. We compared it. So this is a CT scan, pretty much same thing humans will do. But we can see the difference in porosity here. So this orange color is pores. It means air, oxygen in the soil. So CT stands for conventional tillage, no tillage compared to prairie. And here we can also see some of those biopores created by these earthworms and also roots, because are just another important side of biology that we have in the soil. And one of the most often questions I get here in North Dakota is about how long does it take to improve soil health, right? So what does the process look like? And we've degraded our soils for more than 100 years. So what I usually tell them is that soil health doesn't happen overnight. There's a transition period when we move from conventional approaches to more of regenerative or soil health approaches. And everything starts with the biology of the soil. So biological activity is the first thing to be changed. So without this biological activity, there's no carbon turnover, which means transform residue into organic matter to hold more nutrient and water into the soil, improve soil structure, to improve water and air availability, and then maybe maintain increased yields, but at the end of the day, just being more resilient to weather extreme. So this process takes time, but everything starts with the biology in the soil. Now, another frequently asked question is how to measure soil biology. Now that we have 2 hours, we're going to go through all these different soil health biological indicators. We're going to go and discuss them. Of course not, but this is just an idea for you on some of the options that we have to measure that soil biology. And whenever farmers ask me what is the best method to measure, of course, my answer is always very solid and straightforward because we don't have many options, right? And this is one of the things that we still discuss a lot. So we can go from very simple things to super complex things. And farmers, they are always in the progressive ones. They are always interested in learning more about this test. But because we don't have time for that, I just selected a few of the measurements here that I know a few farmers are doing. I know commercial labs are also offering. So when it comes to measuring microbial biomass, so microbial biomass carbon, phospholipid fatty acids, or EOFAMs, so are widely used for academia. Not much by farmers, although a few commercial labs offer these tests across the country. When it comes to microbial activity, soil respiration was usually the most used one. There is a trend of enzyme activities being more measured nowadays than soil respiration, just because of the how easy it is to measure those. And then these are beta-glucosidase, aerosol sulfatase, acid and alkaline phosphatase, and NAG are usually the measured ones. For community structure, phospholipid fatty acid as well. EOFAMs is just a lower resolution compared to genome sequencing using 16S or ITS. But these are some of the options that we have for measuring that soil biology. And I'm going to skip this one because Dr. Wright explained this really well. But one of the things that we do is pretty much to farm soil microbes. We have to manage the biological component, and farmers, they've been implementing practices like the soil health principles to change that biology and manage that biology and maximize that biodiversity. But at the end of the day, roots is the answer for most of the things we have when it comes to soil health. And just to quickly show you some on-farm data. So this is still from my PhD in Kansas. So in this case here, we had a farmer in a wheat soybean sorghum rotations, and that farmer was doing cover crops both in the summer and the fall. So he had winter wheat in the system, which allowed him to do a mix of pearl millets, then grass, sunflowers, radish, turnip, and flax, and followed by a winter cover crop, which was triticale before soybeans, and it was hairy vetch before sorghum. And in 5 years of research, and that was under no tillage, we gained about 0.5% organic carbon. So you can see here, these green boxes are just flat because there was no changes. There were no changes in carbon, but then you see how we gained carbon in 5 years. And this is a very good slide to tell people that it's nearly impossible to increase 1% organic matter or organic carbon in 1 year or 2 years. There's no miracle. There's no such a thing that you can spray or spread to get up to 1%, 2% in a short time, unless you spread manure and then you measure the manure rather than the soil. So then you can see some of those. And this is just another example. So very similar situation on farm research as well. This was a corn soybean rotation with the addition of cover crops. And this farmer also used poultry litter. And in that case, we gained 0.6% organic carbon and 5 years. Just moving on, some of the things that we measured. So microbial biomass, and on this one here is the one with the poultry litter. You can clearly see the effect of poultry litter. So we have these big jumps in here because of that source of carbon. But when we added cover crops into the system, just this purple box here, you can see that it was a little higher than the poultry litter only. Now, when the farmer was still using synthetic fertilizer, you see that these no-till green boxes here, they were just flat. So no-till really important, is the basis for soil health, but it really stopped the bleed, minimizes erosion. But if you want to increase carbon, improve soil health, you need to do more. You need to intensify the system a little bit more. In this case here with cover crops, you see how we were trending up in terms of microbial biomass in that location. Oh, had an animation in there, so sorry. Soil fungi, very similar situation. We had a slight decrease in soil fungi over time without cover crop with the no tillage system. And then again, it was just trending up and increasing the amount of fungi in the soil. And it's important to say here that this field here was conventionally tilled, so chisel plowed and a cultivator, chisel plow in the fall, plowed in the fall, a cultivator in the spring for a long time. And the farmer, they acquired this field, which was a perfect field for us to start doing some research. So we could see these differences. Soil bacteria is just another example of what can be measured on farm. And then enzymes, beta-glucosidase. So, and if you remember after my first figure, when I showed you about the carbon increase over time, you can see that most of these microbes, they are following the same trend or a very similar trend. As we know, carbon drives most of our biology and the soil. And I just want to show you these data here, which I think it's fantastic. So we were looking into the relationship between fungi and aggregate stability. So we know that relationship exists, but we were interested in seeing the effect of cover crops. So can we strain that relationship when using cover crops? You can see that we were able to achieve larger aggregates. So this is the mean weight diameter. So we were able to achieve larger aggregates when we added cover crops. You see this blue shade here and the blue dots here, which we were not able to achieve without cover crops. And this is the side with poultry litter. So we see that there is an effect of poultry litter in soil aggregation because of all that carbon into the system. But when it comes to forming larger aggregates, we really need roots to help to entangle and physically entangle those soil particles together. So at a certain point, carbon is not enough to build large macro aggregates. Some of the other things that we can look into is the microbial spatial distribution and the relationship. And this gets really interesting. I'm an agronomist, so I have a passion for agronomy and crops too. So it gets really interesting when we start looking into the relationship between yield maps and microbial spatial distribution as well. But that will be probably the topic for a different talk. And of this research, one of the things that we did was to evaluate the most sensitive indicators. And then what we found is that in the short term, microbial biomass, arbuscular mycorrhizal fungi, acid phosphatase, aerosol sulfatase, and aggregate stability, which was a little of a surprise. So these were the most sensitive ones to cover cropping in Kansas. And just to finalize here, this is my last slide. One of the other things we started to look at was about the relationship between the biology and the aggregates. So we separated soil aggregates between biogenic and physiogenic formed aggregates. And then here you can see some of the differences that the biologically formed soil aggregates. So you see some crumbles, you see they are round shaped. You see some porosity in between here. You see your roots growing through. And then the physiogenic aggregates that were formed physically, you see they are solid, massive. They are sharp on the edges as well. So these are some of the differences. And then after 37 years of no tillage, we notice a big difference between the amount of these aggregates. So in the no-till system, and it doesn't matter the type of nitrogen management, if it was urea, manure, or no nitrogen. So we had about 60% of those aggregates being formed biologically. When it comes to conventional tillage here, we have only about 30% of those aggregates being formed biologically. So there are some big differences in there. When it comes to biology, there are different aspects of biology that we can always look into. So there's the macrofauna or insects. There's the relationship with that aggregation, and also the microbiome, which is most likely the most studied one. So with that, I hope I was able to finish that in time. It's always hard to put presentations for 15, 20 minutes because we want to tell you about the world, but we have to really summarize everything. So thank you very much for the opportunity.
Awesome. Thank you, Dr. Pires. Fantastic. Great information from both of you. We really appreciate it. We have enough time to go into a Q&A. And I can ask some of these questions, and then some of these came in before, Carlos, before you came online. So these might be geared towards Dr. White, but feel free, either one of you, to answer these questions. So the first question I see is, realistically, can corn be profitable or profitably grown without fertilizer if we maintain or promote all the soil microorganisms?
Do you want me to try first?
Sure.
Okay. Carlos actually might know this better than me, but let me try, Carlos. Let me see if I can. I work with a corn breeder, organic corn breeder, Walter Goldstein, and he's been trying to breed nitrogen-fixing corn and basically microbe-rich corn. And corn that has more endophytes in it, right? And what he finds is that you can grow it without fertilizer. The yields are not as high. With fertilizer, we're really pushing crops and we're maximizing yield. But that's also expensive. Fertilizer is not cheap. And then once you're using fertilizer, you got to use. You'll have to use fungicides probably, and you'll have to use other agrochemicals. So, all that's expensive. So can it be profitable? From everything that I hear, the answer is yes. Now, the yields are not as high, typically they're not as high. The highest yields are the people who try to combine both use of healthy soils and a minimal use of chemistry. But I'm a lab scientist, and I really am going to have to rely on Dr. Pires to answer this in a intelligent way.
Yeah, no, it biologically makes sense. I would just make a disclaimer here. I'm not a soil fertility specialist. So, but when it comes to biology and soil health, it really depends on the price of the nitrogen fertilizer, right? And really depends if who is fighting with Russia, and because that really changes the price of everything. But biologically, yeah. So the more organic matter we have in the soil, the more nitrogen can be mineralized and be released into the system. So there are a few examples of long-term studies, and I really like them because of that, because they can show us how much corn grain we can produce after 38 years of no nitrogen. So I can tell you that in North Dakota, without nitrogen, corn is still putting about 38 to 42 bushels per acre at a zero cost. It's just the cost of the seed, right? I know in Kansas, so we also had a long-term study there. Corn was more on the 50 bushels per acre to 60 bushels per acre, depending on the year, without any nitrogen for more than 35 years. So I think it was 38 or something like that. So it might be possible, but the level of organic matter and how you farm will dictate that process. So just putting cover crops into the system to have that nutrient cycling will be really important. But price of the fertilizer most of the time will tell you if you can be profitable or not.
Okay, I'm gonna sneak in here. It's Heather Nykirk. I'm an extension educator with OSU Extension in Stark County. And so as we look on, Matthew has a question related to how does plant diversity impact the rhizophagy of the rhizophagy cycle, and particularly thinking about the contrast between the diversity of the cover crop mix compared to a monoculture cash crop. So again, Dr. White or Dr. Pires, you guys can tag team these as you see fit.
Thank you. Yeah, the plant diversity is critical, and the reason really is that every plant carries different endophytes. Every plant will cultivate a little bit different profile of microbes. So the more cover crops you have, the more diversity of cover crops, right? The more diversity of soil microbial community. And because it's a kind of a non-specific symbiosis, rhizophagy or rhizophagy, it means more microbes you have, the more microbes plants can get in rhizophagy. Also, there's a certain almost a bootstrapping effect when you have microbes that are growing in one plant and then being put out into the soil, and they go into another plant. So you have a kind of a sharing of nutrients, like they share the microbiology as well. I'm getting kind of old and I can't remember different parts of the question. Was there another part of that question, or did the diversity part?
Yeah, I think the contrast between diverse cover crop mixes compared to just monoculture cash crops. So just standard rotation.
So that was it.
Yep.
Yeah. Carlos, you want to add to that, or?
No, whenever I see rhizo and you are on the call, I'll let you answer that.
Okay. Thank you, Dr. Pires.
Well, we have lots of questions. There's no doubt about that. So we're trying to work through them. You guys got everybody thinking, for sure. Another question came in terms of microbial work in potting soils. So sterile seed starting mixtures, and if not, then how do those particular seeds or plants grow? So I'm thinking this is more of a gardener, like a home gardener or gardening based question. We have a lot of master gardener volunteers who have interest in that.
Well, the seed, I'll say the seed carries microbes, and that's where you really don't want to have a sterile seed. The seed will carry a community of microbes on it, and it's and there's some mixes that suppress microbial growth. But again, the seed carries their microbiology that are critical for early development, and that's really the best answer I can get. There also is no truly sterile soil mix. I mean, we got to work really hard to sterilize it. So there's clean mixes, but is it really sterile? No, it's really hard to sterilize. Even when we autoclave soil or plant material, if there are endospore formers in there, oftentimes it doesn't get the endospore formers. And you can also tell fungi frequently try to autoclave and kill microbes in soil, and then you pull them out of the autoclave and let them sit. Well, you get fungi growing on those soil. It's really hard to truly make them sterile.
Another question from Van, do all plants use rhizophagy? I think we might know the answer to that.
Well, they do at some point in their life cycle, or they can use rhizophagy. The cases where they don't are in very, very acid soils, and the acid soils tend to inhibit bacterial development. But instead, those plants like blueberries or cranberries, they'll get these ericoid mycorrhizae that kind of actually replace root hairs. So I should say any plant that makes root hairs is doing rhizophagy cycle. Any plant that makes root hairs are doing rhizophagy cycle, and that's most plants. Some plants like trees will replace those root hairs, with fungi at some point, and then of course they don't need to do it. They have fungi that kind of substitute for that mycorrhizae, right? Ectomycorrhizae. But most plants do it at some point in their life cycle.
Okay, another question. Is CO2 respiration an indicator of how active rhizophagy and AMF are in soils? How harmful is tillage, pesticides, and salt-based fertilizers to rhizophagy and AMF?
I think it is a reflection. Microbial activity is now how close it is, I can't say, but it does. More microbial activity in the soil, and very high likelihood that you have more rhizophagy or rhizophagy with plants, with crops growing in that soil. And the other question had to do with what, salt or something? Or
how harmful is tillage, pesticides, and salt based on salt-based fertilizers?
Salt. So they would be harmful. I would say salt is one of the key ones. The higher the salt is a problem. The agrochemicals, depending on whether they're inhibiting microbes directly, right, or inhibiting insects, soil mega and microfauna that affect the microbiome, and they certainly do. And the mesofauna and the macrofauna in the soil, they're really important. The insects and so forth, they're really important for creating an environment and spreading insects. The guts, the same microbes plants need, are the ones that are in insect guts. Earthworms are full of the same microbes that plants need. So there's a direct relationship there. Was there something else in there? I'm sure I forgot something else in there. But I
think looking at the relationship that exists between soil microbes and fertilizers, pesticides, things like that. So I think you got a pretty good handle there. Go ahead.
I just want to add a little bit to that one, because soil respiration is a measure of activity, right? It's not about who is there and who they are, what they're doing. So it's a measure of the whole activity of the soil. So we pretty much measure the CO2, carbon dioxide, that is released by those microbes, and that way we quantify how active they are. And there's a very cool research that Chuck Rice did in Kansas. So he actually sprayed fungicide and then measured AMF over time, and there was a considerable decline of applying fungicide in soils and the AMF community. So it definitely has an effect. And then when it comes to tillage, there's a bigger effect on fungi, because it just breaks down all the networks, mycelia and hyphae and everything that is in the soil. However, it promotes fast-growing bacteria as well. So usually after tillage, because of all that flush of carbon into the system, you're going to see higher respiration. So if you measure soil respiration after tillage compared to a no-till system, you're going to see higher rates in the conventional tillage. So some of these indicators have to be used with caution as well.
Excellent points. Very good. Okay, I'm going to hop over to the chat here because we do have some questions that filtered into there. But looking at the type of nitrogen, ammoniacal or nitrate, how does that affect the process? In relationship to probably the rhizophagy, I'm guessing that was during that.
I mean, that's a good question. And I, I can't actually answer that. I think, my general answer is the closer to organic forms of nitrogen, the better. Some forms actually may be inhibitory to the process. We don't actually know. But we do know that from in vitro and kind of studies in the laboratory that some kinds of nitrogen will stimulate the oxidative interaction with microbes, and some forms seem to suppress it. So, but that hasn't been published, and we don't have very much data on that. So I can't say very much more about that. Carlos, I don't know if you want to.
Yeah. Well, there are mixed effects, and it's usually about the rate, right? It's just like any other drug. It's just, well, not any other drug because it's not a drug, but like drugs, it depends on the rate and for how long you do that. So when you have high rates of fertilizer, synthetic fertilizer, and for a long time, you have a bigger effect. So you're going to suppress some of the communities, so the community won't be as diverse as it used to be, and it will definitely promote fast-growing bacteria again, very similar to the tillage process. So it's really about how much you use and for how long.
Nice answer.
Excellent. And then Dr. Lawrence had a question. Can the plant cell differentiate between good and infectious microbes that are entering the root?
Okay, that's a good question. It's hard to know if the plant cell can differentiate, but if it takes them in for them, and under most cases, it can control the microbes. There are some microbes that are highly oxidatively resistant, and they actually become pathogenic when they go in. These are microbes that are, for example, produce their own antioxidants. And because the plant is actually controlling them using oxidative compounds like superoxide, if they're highly oxidatively resistant, the plant can't control them, and so the microbe replicates out of control, and it actually uses the plant as a means of producing their own cells. But in practice, that's what happens in the laboratory. But in practice, even those microbes, when they're put into soil on the plant, don't appear to inhibit. Instead, what we see is a general stimulation. And the reason why we think is that because the plant takes a whole community of microbes in, one microbe that goes in that may be harmful doesn't have that harmful effect, or it may be excluded because the plant is taking in these better microbes. And in terms of pathogens, like, for example, fusarium, that normally could colonize a plant and cause disease, if there's a lot of microbes there, a lot of other microbiology, a lot of bacteria in particular, fusarium is not pathogenic, and so there is this frequent effect of bacteria on pathogenic fungi that reduces the virulence of the pathogens, so that healthy soil, healthy diversity of microbes, then disease agents are suppressed, become less virulent.
Thank
Hey,
you,
I
Dr.
think
White.
we're gonna kind of hold off on answering any remaining questions. I'll kind of toss the baton back to Jim.